Hello, welcome to the video. This is a presentation of the complete theory of evolution and how to eliminate humanity. A brief warning before getting into the presentation proper. After this theory was discovered, it became apparent that an understanding of how to subvert and destroy all of humanity is necessary for a complete understanding of evolution and biology. This presentation means to explain the theoretical basis for ending human existence and providing a broad instructional for how to go about doing so. If you consider the knowledge of how to eliminate human life from the planet to be something you'd like to protect your brain from, then I invite you to leave now and click away from this video. For those of you who have decided to continue watching, a brief disclaimer: this theory was invented by Jean-Francois. I'm sure I've got the pronunciation wrong. It was invented something like 6 to 7 years ago and finally completed somewhere around 1 to 2 years ago. The book explaining his theory was published online on the 7th of December 2018. I should also warn the audience that I may slip in and out of a fake, forced British accent to mask my somewhat indiscernible Australian accent. I apologise to any Australian viewers, I apologise to any British viewers, and I'm of the assumption that American viewers couldn't tell the difference to begin with. This video means to provide a summarized and simplified presentation of the theory of phenotype revolutions. The theory of JFG is effectively an amendment to the theory proposed by Richard Dawkins in his book The Selfish Gene. If you have any issues with what I'm saying or wish to make a challenge, please do so in the comments below or to JFG himself. The scope of this presentation will be very fundamental, as the question being addressed is the origin of life itself. And considering we're dealing with a very fundamental issue, we'll need to start with a very fundamental understanding. When dealing with any claim of knowledge, fundamentally what you're dealing with is the question of what is the case. René Descartes is attributed as saying, cogito ergo sum, or I think therefore I am, a statement extending from the observation that an observation has been made. Any statement or argument made about the topic of what is built upon the axiom that the universe exists. Physics builds upon this axiom to study the components of this universe that is. In addition to assuming the existence of a universe, physics also requires that the universe is both knowable and that it's consistent in order for predictions about the universe to be able to project future events. Biology adds yet another axiom to physics and separates entities into environment and replicator, where the environment is everything in the universe, and the replicator is the component of an organism that is copied through reproduction. The fundamental distinction between physics and biology is that biology relies on the axiom that replicator phenomena exist. Biology, being the study of the interaction between replicators and the environment, raises some questions. What is the media which constitutes the replicator which produces life forms, or human life forms? Are there multiple replicators on Earth? Are there multiple replicators participating in the creation of the human phenotype? What could these other replicators be? But before we can answer these questions, what the heck is a replicator? Replicators have been defined by JFG as entities that make almost exact copies of themselves that also do other things. This definition has 4 components. First, entities, which are things that exist. Next, almost exact copies is used to mean imperfect copying to some degree, with some chance of error or some degree of error. Themselves, meaning that the entity has informational causal access to its copying, something I'll expand upon later. And other things, meaning that they do anything more than existing. This definition is distinct from previous definitions by biologists like Richard Dawkins, who do not include the qualifier of themselves into their definition. I'll spend the remainder of the presentation justifying this distinction, by way of example. There have been many replicator candidates proposed. DNA, cars, RNA, DNA-RNA tangos, digital bits on computers, memes, the universe, fat, protein, dung bowls, the solar system, or even the universe itself. There's a potentially inexhaustible list of entities that we might suppose are replicators. For this example, I will consider the possibility that cars are replicators, and address whether cars should be considered living things or not. Colloquially, people will say things constructed by humans, such as cars, will evolve over time. What makes cars not replicators? Is it possible that cars actually are replicators? And spoiler alert, they may be. If cars are replicators, they certainly use a hive-type system of replication, like bees. So let's compare them. In the bee replicator system, a queen bee produces many worker bees. These bees do not produce progeny themselves because they're born sterile. This makes them the phenotypic machines of the queen bee, which is itself the phenotypic machine of its DNA. Rather than possessing their own reproductive potential, worker bees provide pollen to their queen, and in exchange, the queen perpetuates its own DNA. A parallel to this exists in chicken farms. In a chicken farm, a queen chicken is selected to produce many worker chickens. These chickens do not produce progeny because they're delicious. This makes them the phenotypic machines of the queen chicken, which is itself the phenotypic machine of its DNA. Rather than capitalizing on their own reproductive potential, worker chickens provide meat to humans. In exchange, the humans perpetuate the DNA of the queen chicken. Now let's see what happens when we apply this model to cars. An engineer produces many cars. These cars do not produce progeny themselves because they're born sterile. This makes the cars the phenotypic machines of the engineer, which is the phenotypic machine of its DNA. Rather than having their own reproductive potential, cars provide money to humans, and in exchange, the humans make more cars. There's no line of descent for the cars. But what would it take for the cars to have their own line of descent? And is it necessary? Chickens are generally born with reproductive potential. If they're not made into delicious tendies, chickens can replicate. It's not out of the ordinary to expect that chickens may escape their chicken farm and become replicators in their own right. Worker bees have very low reproductive potential. However, under some circumstances, worker bees will develop into queen bees by eating queen jelly, and then produce their own line of descent. Comparatively, no car has ever been observed to replicate. There sure are a lot of similar looking cars produced with variation though. Maybe cars are tricking humans into making copies of them, like a virus. This could be a viable justification for cars to be considered living. So is there then an important difference that makes cars not living and humans living? Returning to the previous definition of a replicator, entities that make almost exact copies of themselves that also do other things. Cars exist. Cars trick humans into copying them, potentially. Cars do things, and so they may be subject to selection. However, cars are printed by humans. They are a kind of replicator, a full replicator. They replicate themselves in a manner directed by humans, and so are not possessing evolutionary selfishness. What is so important about cars being assembled by humans that make them not living? To understand the significance of the difference between replicators like DNA and full replicators like cars, it is important to first introduce some new concepts. First, a full replicator, the product of a trickster printer, in this example, a car. Trickster printer, an entity that has informational causal access to the composition of some material. In this example, the factory used to make cars. Informational causal access, having the ability to direct the printing of some other information to be a certain way based on the composition of the printer. In this example, the property of a factory which results in the material of the cars it makes being the way they are and not some other way, like a truck. Using these concepts together, we can come up with the sentence that a trickster printer has informational causal access to some material which is then the full replicator of that printer. And so, in our example, a trickster printer, which is a factory, has informational causal access both in its composition and its printing of a full replicator, in this case, the car. Going back to our automotive example, we can better understand why it is relevant that an entity is or isn't a full replicator. The distinction between replicators and full replicators is significant because a full replicator doesn't pay the evolutionary price for its failure to replicate. The printing entity does. The existence of the car is rooted in the existence of the printing entity that produced it. The car cannot violate that process, and so will only ever evolve towards subservience to it. In reality, a car doesn't suffer the evolutionary consequences of its failure to replicate. The engineer does. Cars do not have intergenerational control over the composition of future generations of cars. The composition of new cars evolves towards the evolutionary interests of the humans that control their printing. In contrast, DNA is a replicator since it directs its own printing. If a DNA strand fails to perpetuate, the entity that suffers the evolutionary consequence is that DNA strand. And so we can say, under at least some circumstances, DNA directs its own printing. DNA is the product of self-directed printing, making it a replicator. With this understanding, let's go over some replicator candidates. As far as we know, there are no car replicators, since there's no car that directs its own reprinting. By the same definition, memes also fail to reach the status of replicator, as they're printed by human brains. The dung balls of dung beetles also fail to meet this standard, since they're the product of the dung beetle. There may be a line of dung balls being modified by their interactions with the environment over time. However, these dung balls will never have their own evolutionary self-interest. so long as they're reprinted by dung beetles. And then we get to DNA, which is a replicator in at least some circumstances. But why some? Why only some DNA? Well, we have a definition for a replicator, and we know why something is a replicator. It self-copies. So then, this allows us to ask some questions of DNA. Does all DNA qualify as a replicator? Do proteins encoding genes qualify? What about viral DNA that has managed to insert itself into a DNA genome? What about mitochondrial DNA, which performs a cycle of copying external to genomic DNA? What about telomeres, free-floating nucleotides, satellite DNA, or non-coding regions? All of these things are demanding definitions of their borders in order to determine whether a particular segment of DNA qualifies as a replicator. And so we must first ask, what is the DNA replicator unit? This is a question that's been proposed by biologists since before the discovery of DNA. Early biologists proposed that the species was the fundamental unit of evolutionary selfishness. However, this concept is violated by a single individual that has greater reproductive success by defecting from its species. The genome, then, as the smallest selfish component. Well, no, since the array of combinations of chromosomes existing in different people may propagate at different frequencies, and so evolutionary pressure may be applied to different chromosomes to different degrees. The chromosome, then? Well, no, since transposable elements may shift around within a genome or within a chromosome and propagate themselves at different frequencies to that of their native chromosome. The gene itself can also be divided into intron and exon, coding or non-coding. These components may also be subdivided, and so even these subcomponents within a gene may have distinct evolutionary fitness. We can continue refining this process down to the base pair, which has a single base of DNA and a complementary matching base on the opposite strand. However, even in this situation, if one of these bases are to mutate, DNA repair mechanisms will have to make a decision about which base was in error and replace the other. And so even the complementary base in a nucleotide base pair may have a distinct evolutionary self-interest to its complementary base. But we can go no further. The base itself cannot be subdivided without losing its capacity as a replicator. And so by this metric, we can say that the base is the unit of evolution for DNA-based genetic media. Again, Mr. Gary P. has defined the unit of inheritance of a replicator as a queen. This definition is in line with the nomenclature invented by Richard Dawkins, e.g. gene, meme, queen. A queen is defined as a quantum of inheritance, the discrete, irreducible unit of inheritance of a replicator, capable of making copies of itself within at least a certain environment. Now that we have a definition for the unit of inheritance and evolutionary selfishness for DNA, what queens qualify? Let's compare some regions within a hypothetical DNA-based genome. This genome has random junk DNA, satellite DNA, which is a region of DNA with thousands upon thousands of repeats used as an anchor point for chromosome separation. The ribosome complex, responsible for making the proteins of a cell. DNA polymerase. When evaluating the replicator status of the bases of junk DNA in a genome, it's immediately apparent that junk DNA is not directing its own reprinting. Reprinting is done by DNA polymerase. And so despite being composed of DNA, there is no informational causal access for junk DNA to the printing of its copies. The same can be said of satellite DNA, which, though possessing a known function, does not direct its own reprinting, which is the same criterion by which ribosomal DNA doesn't qualify as a replicator. Out of these candidates, only DNA polymerase qualifies as a replicator, as it possesses information that directs its own reprinting. DNA polymerase, and all of the subunits that compose all of the polymerases, direct their own printing, in addition to satisfying all of the other requirements of a replicator. So now that we've refined the replicator component of a genome to the subunits of DNA polymerase, and we've already defined the queen of DNA as the base, we can question whether all of the bases of DNA polymerase are as selfish as each other. Let's compare some regions within a hypothetical DNA-based polymerase gene. Let's compare the coding regions and the non-coding regions. Despite being part of what is classically considered the DNA polymerase gene, introns do not direct their own reprinting. Only the exons, which are the bases of the gene which are translated into protein machinery, direct their own reprinting. The important property of these bases, in terms of their being replicators, is that they code for machines, in this case proteins, that direct their own printing. And this leads us to the completed theory of evolution. For DNA, the individual bases of DNA which have informational causal access to their own printing constitute the DNA replicator. I refer to the ensemble of these bases as DNA replicase. They are the only component of a genome that is subject to evolution. All other segments of DNA are the full replicators of those bases. Mutations in those full sequences of DNA are significant to the evolution of DNA replicase to the extent that they influence the fitness of DNA replicase. It can be understood that Richard Dawkins is therefore wrong in saying that genes are selfish. In truth, they're entirely selfless, with the exception of DNA replicase, which remains entirely selfish. That's amazing! That's amazing! This is an amazing theory! Be amazed! You! Amazed! Amazed! If you do understand it, pat yourself on the back. This leads to some very fundamental questions about the history of DNA, particularly, how did DNA gain its replicator status, and how will DNA lose its replicator status? On the question of gaining replicator status, there are two hypothesized paths to this genesis of evolutionary self-interest. Abiogenesis, evolved self-interest from non-living components, and the newly proposed theory of JF, a phenotype revolution, which is evolved self-interest from the phenotype of another replicator. So what is a phenotype revolution and how does it occur? A phenotype revolution is the product of a replicator tango. A replicator tango is the transitionary period where two distinct replicators are each printing each other, each containing the gene for the other's replication. It has been established that RNA was almost certainly the genetic medium of our phenotypic, e.g. non-genetic ancestors, and replicator tangos explain how this occurred. Either through independent development of a DNA polymerase or viral insertion, RNA-based life began generating DNA. DNA was useful to the RNA replicator, which gradually exported gene copies into DNA. DNA was then used as a template for printing future copies of RNA. Eventually, the DNA acquired a replicase gene from the RNA genome, or from random. DNA was then used as a template for printing future copies of RNA. This initiated a replicator tango, as the reprinting of this entity was now passing through both RNA and DNA. We now have RNA replicating with error, and DNA replicating with error, as well as RNA and DNA both having some capacity to print the other's genes. These replicators are now both evolving within the cell, attempting to farm or print the other replicator towards its own fitness. As it turns out, DNA was the more fit genetic media. Eventually, RNA abandons its replicase gene, and so its capacity for evolutionary selfishness, as all printing of the RNA will be done by an external media. DNA could then print RNA with impunity, as RNA's composition must evolve towards the selfish evolutionary interests of DNA, as the car does the engineer. DNA won this evolutionary race, and has since covered the Earth for about 4 billion years, while continuing to selfishly print RNA. An understanding of this theory demands a revision of phylogeny. This is the conjunct history of the origin of DNA-based life. In the revolutionary phenotype, the author proposes an amended method of denoting ancestry to include reference to phenotypic, non-genetic ancestors, as shown here. In other words, the significance of this is that humans have ancestors to which they can draw no genetic lineage. The complementary question, how will DNA lose its replicator status, leads us to asking some interesting and worrying questions. How might the next revolutionary phenotype come about? What might be the consequences for DNA-based life? Has DNA-based life already created a revolutionary phenotype? And now finally an answer to the question, how do you end humanity? There is no great likelihood of a phenotype revolution occurring from genetic modification directed by humans. This is true whether the genetic alteration is directed by sophisticated protein machinery, in the case of DNA repair, or by crude gene modification techniques. We already describe genetic modification as a mutation event, and this is accurate. Humans have a fitness requirement for computing power. This requirement for computing power is partially satisfied by the development of a brain. This biological computer will be largely responsible for the directed mutations scientists do in the future. Could the brain be the instigator of a phenotype revolution? Well, brains are a kind of plastic computer, meaning their composition is altered by acquired information, and the cells also replicate and mutate. However, viewed as organisms, they have a fitness value of about 0. Brains can't propagate to the next generation, and so the evolutionary interests and the structure of the brain, and all the ideas it encodes, are subjugated to the reproduction of human germline cells. A brain probably won't revolt against DNA. What can? Humans have discovered a greater fitness benefit in creating more computing power in the form of digital computers. We store copies of our genes on these computers and, if staying our current course, we'll use them to direct mutations in humans, e.g., to print human genes. We store the instructions for creating computers on computers. In this way, computer phenotypes, such as the Dell phenotype presented here, can reproduce their digital genome by recruiting humans to reproduce them. Digital replicators may emerge as a life form on Earth if humans don't continually intervene in their code. This eventuality where digital replicators separate from humans is not so significant for human biology. In this event, digital life forms may result in another competing life form on Earth, like RNA viruses. The more significant event for human biology is the gradual gain of control of human gene printing by digital replicators. Inviting digital replicators into the process introduces a serious possibility of a phenotype revolution. Digital replicators evolve independently of DNA through random mutation or self-modification algorithms. Using these evolving digital replicators provides a competitive advantage to the DNA life forms they engineer. If digitally directed DNA modifications continue to provide a fitness advantage to humans, they become increasingly preferred for human reproduction. Simultaneously, digital replicators evolve to promote their own propagation, including the modification of humans to serve the digital replicator, if only a little at first. As the digital replicator provides greater benefits, the selfishness of human DNA is directed towards preservation of the computer. These life forms are eventually commandeered to pursue replication of the digital life form over their own, as the digital life form becomes increasingly responsible for the reproduction of human genes. DNA then ceases to be an independently evolving replicator, and is now a subjugated molecule to the evolving digital bit. DNA-based ancestry ends. What can we do to promote a DNA-to-bit phenotype revolution? Well, we could spend millions to billions of dollars in a global effort to develop self-modifying intelligent computer programs. We could then allow these programs to influence our behavior in a way that affects the fidelity of gene copying, e.g., computer-directed gene printing, as we are busily doing today. What can we do to prevent a DNA-to-bit phenotype revolution? Well, we can decouple self-modifying digital replicators from any influential role in printing the DNA of the life forms that make the digital replicator. We can also periodically purge digital replicators to prevent their successful adaptation to life on Earth. And we can preserve the knowledge of the threat posed by revolutionary phenotypes, as this knowledge is requisite to subverting the evolutionary process that would end life on Earth. Now, I'll make a call from my personal intuition. We're not going to do it. Soon we're going to see the initiation of a printing process directed by an external informational media applied to humans, and so the start of the end for humanity, and consequently the vast majority of all DNA-based life on Earth. We had a good 4 billion year run. Instead, DNA will become another layer of information transfer in the chain from replicator to phenotype. At this point you may regret having watched the full video. Being on the cusp of the impending replicator tango, we have the unique opportunity to recognize the approaching demise of humanity and to care to stop it. In any case, thank you for listening to this presentation. I should inform you that this is an incomplete representation of the works of Jean-Francois. You can find a link to his YouTube channel below, as well as to his amazing book, The Revolutionary Phenotype. Within those resources you'll find much better explanations and extrapolations of this theory.